Thank you very much for that introduction, and thank you very much to the organisers. It's always a great pleasure to come to Bangkok for this very high-quality conference. And you know, I think the presentations overall have been of outstanding quality so far, and I hope I can keep up the standard. These are my potential conflicts of interest. So, as we've heard throughout this conference, and as we know, antiretroviral therapies have really revolutionised both HIV treatment and prevention, and particularly around treatment. Although it has transformed the lives of millions, the treatment simply holds the virus in check. It allows immune reconstitution and immune preservation, but it doesn't clear the virus. And treatment is for life, because we know that as soon as we stop antiretroviral therapy, the virus comes back, you get viremia and disease progression, usually within weeks of stopping antiretroviral therapy, even if you've been on suppressive therapy for many, many years. And this rebound comes from a reservoir. And so what I want to cover today is an update on what we understand about this reservoir, and how people are trying to manipulate the reservoir to drive forward the holy grail of cure and remission strategies. So the challenges of HIV control are in many ways similar to those of HIV treatment. The virus is a challenge. It is highly diverse, and any intervention has to accommodate the diversity of the virus. Not only is the virus diverse, but the cells in which this reservoir exists is also diverse. And so many cell types harbour the virus that makes up the reservoir, and these include CD4 T cells, particularly resting CD4 cells, but also include cells in the bone marrow like hematopoietic stem cells, and then cells of the myeloid lineage, such as monocytes and macrophages, and cells in the CNS, such as both microglia and astrocytes. So those cells are disseminated throughout the body and are found in multiple organs. They're predominantly in the lymphoid tissue, such as lymph nodes, spleen, liver, but are also in sites that are more difficult to access, particularly the brain, behind the blood-brain barrier. And then the other problem is that the reservoir is not, although it's there and highly problematical, it's not very extensive. And so across all these sites, only about 1 in a million cells are harbour latent virus. And so you have to come up with something that is going to be able to get to all these sites and affect these relatively rare cells. And then anything that you produce has to be accessible and affordable in an equitable basis. So the challenges are considerable. So just some background on the reservoir. The reservoir is made up of HIV in its DNA form, and there are various ways of measuring HIV DNA, and there are various forms of HIV DNA. We can measure total HIV DNA. We can measure that part of the HIV DNA which is actually integrated into the chromosome, which we know needs to happen for it to be able to reproduce itself. And then only a subset of that integrated DNA is actually full-length and replication competent. And so, depending on the measurement you use, you can get different ideas of the reservoir. But the reservoir that is most important is about 0.1% of the total HIV DNA that you can measure, and that's the replication integrated HIV DNA. So there has been a lot of work done on mapping the reservoir, and the summary of all that work is, as I've said, that it's disseminated and widely dispersed. But the cells, the latently infected cells that sequester fully competent virus are relatively rare. It's found in CD4 CD4 T cell subsets, and it's preferentially found in certain subsets of CD4 T cells, like follicular T helper cells, which are found in the lymph nodes. And increasingly, there is reproducible data that effector T cells carry a greater proportion of this reservoir than other T cell subsets. But this is only relative. No matter which CD4 T cell subset is your favorite subset, you can find HIV in that subset. So this is not an all or nothing phenomenon. Although there is some partitioning of the reservoir, preferential partitioning of the reservoir in these cell types, it is by no means absolute. And this has meant that actually understanding we don't have a simple marker that tells us whether a CD4 T cell is infected or not. You have to lyse those cells and you have to measure the DNA to actually identify whether the cell is infected or not. So there is no simple way of, by looking at a CD4 T cell down a microscope or in a flow cytometer, telling whether it's infected or not, and particularly whether it's latently infected or not. And so that makes estimations about who you should interrupt and who you shouldn't interrupt with analytical treatment interruptions, and actually identifying the cells you should target with interventions very difficult. There is now, I think, a greater understanding of the relative contribution of clonal proliferation, that is a particular CD4 cell responding to a particular antigen expanding with latent virus on seeing that antigen repeatedly, versus new rounds of infection causing new infection during antiretroviral therapy. The relative contribution of those phenomena is getting better and better understood, and we're understanding that the clonal proliferation is probably the main cause of sustained the sustenance of the reservoir in people that are actually stay on antiretroviral therapy and are compliant. There's also been a lot of argument about whether macrophages and monocytes play a role in the long-term reservoir. And I think one of the big changes over the last 5 years is that I think that the dogma that they don't play a role has changed, and there is now a greater acceptance that we have to be concerned about the monocytes and macrophages as well in any intervention that affects the reservoir. And increasingly, we're understanding that that a lot of the virus that is circulating is in fact defective. So what about this latent state? What does that mean and how might you manipulate it? So this is HIV in its DNA form. It's integrated into the host chromosome, but it's not transcribing. So it's not making RNA, it's not making new copies of itself. Gene expression from HIV is driven by a promoter, just like our genes are driven to expression by a promoter. And in the case of HIV, the promoter is part of the genome of the HIV, which is called the 5' LTR, or long terminal repeat. There are various mechanisms of latency, the most important of which is probably this epigenetic silencing, which I'll describe in more detail in a moment. But this involves changes in the histone architecture of the 5' LTR and relies on the acetylation and methylation state of the histones associated with the 5' LTR. The virus can also be latent because of the way where it integrates, that is, that it integrates into silenced genes in the host genome. And so it's integrated into closed chromatin, and that closed chromatin will not express virus. Or it might be silent because it in fact is defective and not replication competent and unable to transcribe. So as I said, the most important contribution to and the manipulatable form of latent HIV is determined by this phenomenon called epigenetics, and particularly around this, the role of histone methylation and acetylation. This is a cartoon of the HIV genome with the 5' LTR shown here and the various structural genes of GAG, POL and envelope shown here, and the other accessory genes. and the 5' LTR is blown up here. And the main things to understand about the 5' LTR is that it regulates transcription, and it does that by binding host transcription factors such as NF-kappa B and SP1, and those host transcription factors activate the virus. And there are a series of host transcription factors shown here in these coloured ovals that the virus can bind, and when a T cell gets activated, it overexpresses those transcription factors because it needs that to turn its own gene expression on, but the virus hijacks those transcription factors to turn itself on. The virus also is transactivated by one of its own proteins called TAT, which binds to a region here. But the other important part of this structure is that there are these two nucleosomes that are associated with the 5' LTR, and these are called nucleosome 0 and nucleosome 1, or nuke 0 and nuke 1, and these are made up of histones. And the methylation and acetylation of components of these histones determine whether you have open or closed chromatin and whether transcription can occur or not. So when you have open transcription, the nucleosomes are relatively far apart, the transcription factor binding sites are accessible, the loop that binds TAT in the virus DNA is accessible, and transcription can start. When you have closed chromatin, the nucleosomes are packed together, the DNA is packed together, and the transcription factor binding sites are no longer accessible, and TAT cannot bind, and you get no transcription. And as I said, this is regulated by methylation and acetylation of various components of these histones. So what's required for a cure? Well, we have some evidence from people that have been treated for hematological malignancies while being HIV positive with bone marrow transplantation. And there are now 4 reports of adults who have been cured of HIV using this procedure. So what are the common features? Well, they've all had significant chemotherapy that is ablative. So they've had their own immune system ablated. And then they've had transplantation, allogenic transplantation. So they've been transplanted with someone else's hematopoietic stem cells. But those have not only been HLA matched as you need for bone marrow transplantation, but they come from a donor that is homozygous for the delta 32 mutation of CCR5. So they don't express a functional CCR5 receptor, which we know is the co-receptor for HIV. So their immune system has been reconstituted with CD4 cells that are resistant to infection by most strains of HIV, or at least CCR5 using strains of HIV. So this gives us a hint of what we need to achieve for cure, but it's not a viable option for people without malignancy because, one, the source of hematopoietic stem cells that are HLA matched and CCR5 deleted is simply very rare. It's a very expensive procedure, and I think most importantly, the morbidity and mortality associated with this procedure is very significant. And often this bone marrow transplantation is not even the first-line therapy for these hematological malignancies and really is rescue therapy. So there is significant morbidity and mortality associated with this approach. So can you mimic this with gene therapy? So certainly people have tried to knock down CCR5 using various approaches, various constructs such as zinc finger nucleases, TALENs, and CRISPR-Cas, as Sharon was talking about yesterday for COVID. And these can be these have been transfected into CD4 T cells or into CD34 hematopoietic stem cells. So you take cells out of the body, you transfect them with these gene constructs that express one of these components that knock down CCR5, and then you reinfuse the cells into the patient and hopefully they engraft. So in summary, these have been phase 1 trials. They've shown limited effectiveness because there's been rapid recrudescence. And the problem is, what you need to do is make sure that every CD4 T cell in the body expresses a knocked down CCR5 phenotype. And at the moment, the efficiency of the delivery, the efficiency of the transcription of the transduction of the gene therapy constructs, and then the engraftment and implantation of those cells into the bone marrow and their ability to repopulate the immune system is too limited. And so there is always a source of cells, CD4 cells, to be infected by the virus. And so the virus wins and gets its upper hand. So you get this transient reduction in virus and the reservoir, but it's short-lived. So what other approaches have been taken? So there are 2 major approaches that people are looking at. One is summarized as shock and kill, where you activate the virus in people who are on antiretroviral therapy. You try and wake up that latent virus, and then you try and kill the cell that is infecting that is being expressed by that latently infected virus. So there's a range of approaches that have been undertaken. These include drugs that manipulate those epigenetic profiles, those histone profiles. So you change the acetylation or methylation profiles of those histones using HDAC inhibitors or histone methyltransferase inhibitors, or you activate the T cells using PKC activators. And then you try and kill that cell, usually with the host's immune response through cytotoxic T cells, seeing the antigen being expressed by these cells. And I'll go into a bit more detail on that in a moment. The other major approach is what we call block and lock. So rather than trying to clear the reservoir, what you try to do is lock the virus in its latent form. And again, this is by manipulating the epigenetic profile of the virus using things like shRNAs or siRNAs or TAT mimetics such as TAT now basic or some small molecule inhibitors. And what you're trying to do is induce something like super latency where the virus cannot wake up. And you really, I guess in that approach, you're trying to mimic what has happened to many other retroviruses that populate our genomes. There is a lot of retroviruses in all our genomes that are silenced and that we live with and that we've evolved with over the eons. So a bit more on the advances on shock and kill over the last 5 years. So there's been a range of latency, over the years there's been a range of latency reversing agents used, including HDAC inhibitors, various cytokines and PKC activators. There's a growing realization that these need to be used in combination because most of the single agents, when they're used, result in only modest reactivation of the virus. Last year there was a great paper published in Science Translational Medicine that did a full genome screen looking for new LRA targets that hopefully will produce some more targets for us to look at in terms of shock and kill. The issue of the non-specificity, the problem with a lot of these approaches is that you manipulate the epigenetics of the virus, but you also potentially manipulate the epigenetics and therefore the regulation of many other host genes as well. And so there's a lot of off-target and potential toxic effects. And the issue about using these things in combination is that you will potentially get increased toxicity, and some of that toxicity might be unexpected or unpredictable. And a good example of that is, again, a paper published from Sharon Lewin's group last year where they ran a trial of using varinostat with disulfiram. And they got unexpected neurotoxicity from that combination that means that combination can't be used. And targeting of these LRAs to try and reduce these off-target effects, so you're just targeting the latently infected cells, is difficult because, as I told you at the beginning, the reservoir is very disseminated and we don't know what a marker is of a latently infected cell, so you can't make a nanoparticle that will just target the latently infected cells. So we need to get better at managing these off-target effects. With the shock part of this, with the kill, what we know is that simply waking the virus up and allowing the natural immune response to kill the targets is not enough. It has to be boosted. There is increasing interest in using PD-1 and CTLA-4 checkpoint inhibitors to boost this immune response, as we do in cancer. There is potential proof of principle of this in a paper published 2 years ago by Thomas Rasmussen, where he looked at reservoirs and people being treated with these agents who had cancer, and he looked at the reservoir, and in some of those patients there was a potentially reduction in some of the measures of the reservoir in those people that had received PD-1 and CTLA-4 inhibitors in combination for the treatment of their cancer. There is also new work on NEF inhibitors to NEF downregulates the ability of the immune system to see a virally infected cell by downregulating MHC1, and there's a new class of NEF inhibitors that may improve this, and there are new advances in the old therapeutic vaccination of networked conserved epitopes that may boost the immune response and help clear the reservoir. There has been the use of broadly neutralizing antibodies to try and clear the reservoir in a randomized controlled trial. What this shows in these individuals who received recurrent doses of these broadly neutralizing monoclonal antibodies, that there was no change in any measure of the reservoir in the people receiving the monoclonals. And that not all viruses, even though these are broadly neutralizing antibodies, they're not broad enough. There's lots of the virus that aren't actually neutralized by these viruses. So it looks like if these are going to work at all, they need to be used in combination. There's also a lot of work being done on CAR T cells. That mimicking what's been done in cancer, we need to know much more about the epitopes and the T cell receptors to design those CAR T cells that may help clear the reservoir. What about block and lock? So this is really dependent on gene therapy approaches. One of the approaches we've undertaken is using the shRNAs, which bind to conserved regions of the 5' promoter. Prom-A is one of our constructs. This is a highly conserved sequence across many of the clades of HIV. It binds to that sequence and then causes chromatin compaction that lasts for many weeks in vitro, and which causes a 2 to 4 log reduction of virus in vitro. This causes epigenetic changes, which I won't go into here, but those epigenetic changes are resistant to reactivation by LRAs, such as TNF and varinostat. And so these are cultures treated with various doses of TNF and varinostat, and the cultures treated with the Prom-A or with one of our other targets don't wake up despite the use of these very high doses of the LRAs, and these are the doses at which they should wake up, and even at much higher doses they don't wake up. And then when we use these constructs in mice, we get suppression of virus and preservation of CD4 T cells. So promising, but in an acute model of HIV. So what we've done more recently is tried to combine approaches where we take the gene therapy approach of knocking down CCR5 and the gene therapy approach of this block on lock using our PROM-A targeting the 5' LTR. And what this idea is that the cell is both has silenced virus and is also not prone to reinfection by any virus that's not silenced because we've knocked down CCR5. So what we tried to do in humanized mice is mimic how this would be used in humans. We gave the mice the construct, we gave them HIV, we treated the mice with antiretroviral therapy, we put them, we got their viral load down to undetectable for several weeks, and then we interrupted and saw what happened. And we had various groups of mice, some treated just with PROM-A and some treated with this combination in green here, and we had the controls in black and blue here. The take-home message is that the combined therapy maintained CD4 T cells in these mice, that there was preferential outgrowth of the CD4 cells that were actually transfected with this, and so were preferentially protected. And there was a modest but significant delay in viral rebound in both the PROM-A and the combination treated group. So early promising data, but we need to get much better at this because essentially the same thing has happened as I described with the CCR5 gene therapy in humans, in that we just don't get enough of the cells carrying these constructs, and the cells that aren't protected by these constructs provide a source for the virus to grow in and reconstitute the reservoir. But this does work not just in the peripheral blood, but knocks down the virus in the tissues. So these are the controls here, and the amount of virus shown in these little silver circles here in lymph nodes. And you can see this is one of the controls and another one of the controls. And then in the combined group, there's far fewer cells that carry virus, and the amount of virus that's being produced, which is reflected by the size of these cells, is also reduced. So not just working in the periphery, but working on the tissues where the reservoir is. So our next steps with this are trying to optimize the selection of the cells that are transfected so that we get 100% of the cells transduced, and to use multiplexed sRNAs that will cover the variability of the virus. And we know we have 4 different targets that are highly conserved, and we know from looking at the database that this will cover 99% of the variants of the 5' LTR. So what are the other approaches that are going on in block and lock? There are small molecule inhibitors like dihydrocorticostatin, which give which will sustain latency for short periods, and there are TAP mimetics that also may achieve this. The most recent data comes from an adeno-associated virus-delivered CRISPR-Cas that will cut the HIV DNA, cut a big section out of the virus, and at least in macaque models of SIV, this has shown reduction in HIV DNA in 3 different macaques from about 30% to 92%. But they didn't interrupt to see what would happen to those macaques. Although 92% sounds very impressive, I think we need to remember that AZT reduces virus by 90% as well, and we need to do much more like 99.9% than 90% to get. So another 2 orders of magnitude before we're going to have success. So there's a lot of work looking at how to get better transduction and better uptake of these cells, either by this sort of taking the cells out, transfecting them outside the body, and then reinfusing them. There's a lot of work going on in that space, but this is very expensive and needs high-end technology. There's a lot of work going on looking at how to package the gene therapy either in viruses or in nanoparticles like lipid nanoparticles that we all know so much about, and giving those directly intravenously and having those being taken up by the CD34 cells or the CD4 T cells in vitro, and so the in vivo, so the transduction happens in vivo rather than outside the body, which is potentially cheaper. We need to get better at targeting the CD4 T cells and getting these things taken up by specifically at least CD4 T cells and not other cells. And there's some encouraging work using various variants of HIV envelope that will increase take-up both into T cells and into monocytes, and also the use of broadly neutralizing antibodies to target infected cells. So where are we up to? So in vitro examples of cure are exceptional and require significant manipulation of the immune system, both in terms of ablation and reconstitution. All the proposed interventions that I've told you about are potentially expensive and technically challenging and have associated potential toxicities, and none have progressed really beyond phase 2A in humans. But there is a significant pipeline and an increasingly sophisticated understanding of the biology. So it's not around the corner and more work's required, but there's a lot of good work happening in this space. And hopefully in a couple of years, I can give you a better, a more optimistic view of where this is up to. But if you really want to hear where the state of the art of this is up to, please come to Brisbane. Please come to the HIV Cure and Immunotherapy Forum, which happens on the weekend immediately before IAS in Brisbane. And for those of you who are interested, the abstract submission closes on the 31st of this month, and I encourage you to submit something. Thanks very much.